Okay, everyone, let's start talking about 10.1 and 10.2, which is just an overview of muscle tissue. All right, so we've seen this before, especially if you're one of my sections that has a lab. We went over histology. We learned about the 3 different muscle types. So again, the 3 types of muscle tissue are skeletal muscle, cardiac muscle, and smooth muscle. So this is something like skeletal muscle would look like, cardiac muscle, and smooth muscle tissue. So the main function of each one of these 3 is generating muscle tension. So that's a basic function common to each one of these. However, you do have more like specialized functions that may be specific to skeletal muscle or smooth muscle, and some of those include, for example, creating movement, maintaining posture, stabilizing joints, generating heat that you kind of typically associate with skeletal muscle. You could make a case for creating movement being cardiac muscle tissue because that's going to help to pump the blood throughout the body. Also, another function that is specific to muscle tissue is helping to regulate the flow of materials through hollow organs. With that one, you typically think of smooth muscle tissue. You think of like peristalsis. You could, again, make a case for cardiac muscle tissue because, again, you're using that cardiac muscle tissue. It's found in the heart, and it's going to be helping to pump basically blood to every place in the body. All right. So again, I know we already talked about this, but this should be a good little review for y'all. So remember, skeletal muscle tissue is striated. So it's made up of long parallel muscle cells. And these are the striations here, these little lines. All right, so most of our skeletal muscle is found attached by connective tissue to the skeleton. And again, our main function of this skeletal muscle tissue is movement. You're going to tell your arm, okay, I need to move this object here. So that makes sense. And again, skeletal muscle tissue is voluntarily controlled. That means you have to think to your bicep, okay, move now, move now. You don't have to rely on, automatic functions to do it. You can actually think, oh, okay, I need to move my arm. All right, so cardiac muscle tissue, again, it is also striated. So again, it has those little lines on there, and it is found only in the heart, hence the name cardiac muscle tissue. So these cardiac cells are highly branched, so they kind of Y or V off. They're highly branched. And compared to skeletal muscle, which is multinucleated, having many nuclei per cell, cardiac muscle tissue tends to be uninucleate, having one nucleus. However, it can have 2. It just depends. You typically consider uni basically nucleated, only 1 nuclei per cell. Cardiac muscle tissue, however, does have very specific junctions. So these are called intercalated discs. So they're going to be joining one cardiac cell to another, and to another, and to another. So these are basically specialized gap junctions and desmosomes that allow the heart to contract and function as a whole. So it helps to coordinate their contractions. Cardiac muscle tissue, thankfully, is involuntarily controlled. I don't have to tell my heart, okay, beat now, because again, I'd be dead in about 5 minutes because I'd just forget. I even forget to breathe sometimes. So it's a good thing that that's involuntarily controlled. All right, so smooth muscle tissue is kind of different than the other 2. Still very important, but it is a little different. So smooth muscle tissue has no striations. These are typically long and flat, kind of spindle-shaped cells with pointed ends. And they typically are uninucleate. They have a single centrally located nucleus. They're also found lining most hollow organs, for example, the skin, the eye, and some ducts. The cells are linked to one another via gap junctions, and again, that allows for synchronized contraction, like for example in the gut with peristalsis. Again, smooth muscle tissue is involuntarily controlled, so again, you don't have to think about this. This is something that automatically happens without you having to physically think about it. All right, so you need to know all of these definitions. I will probably ask you about one or two of these. All right, so they're very, very important, and especially when you get to later on, when you get to the digestive system and when you get to cardiovascular system, these are going to come back into play. So might as well learn them now. Those are all AP2, but still, might as well learn them now. All right, so contractility is the ability of proteins in a cell to draw closer together, to the ability to contract. That makes sense. That was an easy one. Excitability is the ability of a cell to respond to a stimulus. Okay, makes sense. Conductivity is the ability of a cell to conduct electrical changes across the entire plasma membrane, so conductivity. These last two are the hard ones. Those are the ones that people commonly get messed up, and they're the ones I commonly ask. So extensibility is the ability of a cell that allows it to be stretched without being ruptured. So extensibility is essentially being stretched, but we're not going to break. Elasticity, don't confuse the two. Elasticity is the ability of a cell to return after being stretched. So it's the ability of a cell that allows it to return to its original length after it has been stretched. Again, you need to know at least the last two because I typically ask about them. All right, so myocytes or muscle cells, and you'll learn later on there's a lot of different names for muscle cells, so you might want to make a little note for yourself. Myocytes, muscle cells, same thing. They have specialized terminology. They're basically the same things, but anatomists, for whatever reason, like to make up a lot of names for the same thing. So the sarcoplasm is a muscle cell's cytoplasm. It's basically the same thing, but we're going to give it a special name. So you need to know what these are. Sarcoplasm is the cytoplasm of a muscle cell. Sarcolemma is the myocyte's plasma membrane. So outer portion, the plasma membrane. Sarcolemma, the outer portion. Sarcoplasm, all this nice little jelly stuff in here. And the sarcoplasmic reticulum, SR, what do you think that is? And ER, modified endoplasmic reticulum. So it's a little bit specific when you talk about muscle cells because they have specific function. So these are going to form a network around the myofibrils, which are basically bundles of protein on the inside of our muscle cells, which allow us to make our actual function work to contract. And it is a little bit different when you talk about cardiac muscle or when you talk about smooth muscle. It's a little bit different. But for right now, what we're looking at is our skeletal muscle cell, what they typically look like. All right, so myofibrils are cylindrical organelles which are found in each of the three muscle cell types. So these little bundles of protein, basically, are called myofibrils. Okay, so there's hundreds to thousands in each myocyte in each muscle cell, which make up around 50 to 80% of the cell volume. So you would think they're very important, which they are. They allow for our muscles to actually work, perform their functions. So they're around 1 micrometer in diameter, so about 1/100th of the thickness of a human hair. And again, they're essentially bundles of protein. They're made up of bundles of specialized proteins that allow for contraction, which again, we're talking about muscle. That is our main function. All right, so let's talk about 10.2, structure and function of the skeletal muscle fibers. So again, lots of different names for the same thing. Your book doesn't tell you that the muscle cells of the striated muscles are referred to as muscle fibers. Same thing. That's just muscle cells, muscle fibers. It's the same thing. Just so you know, because we're going to mention it, I don't want you to get confused. So make a little note of that. All right. So skeletal muscle tissue consists of many fibers and their surrounding endomysium or extracellular matrix. So skeletal muscle fibers are a little bit different when you're actually looking at them. They're actually kind of like thin cylinders. Okay, so they're up to 30 centimeters in length, whereas in comparison, the red blood cell is only 7.5 micrometers in diameter. So let's just look at this. This would be your skeletal muscle here versus a red blood cell. So quite large in comparison. And this is one entire cell. Pretty neat. All right, so each fiber is surrounded by a sarcolemma. Again, a cell membrane. When you're talking about muscle cells, it's very specific. Sarcolemma, cell membrane, plasma membrane, it's the same thing. So most consist of myofibrils, which are made of contractile proteins. Again, we'll talk about the terms here in just a minute. You also have a sarcoplasmic reticulum, which surrounds the myofibrils and stores and releases calcium ions, which is going to be important when we actually are trying to elicit a muscle contraction. We'll come back to that. So you also have transverse or T-tubules. I'll refer to them here on as T-tubules. That's common. These are inward extensions of that sarcolemma that surround each of the myofibrils. So these are going to form a tunnel-like network which is continuous with the exterior of the cell and are therefore filled with extracellular fluid. Okay. So these are your T-tubules here. This kind of tannish portion in here. T-tubules. And here. So terminal cisternae are in large sections of the sarcoplasmic reticulum, which are found flanking each tubule, so on either side. So two terminal cisternae, here and here, and in between that T-tubule we just talked about, that is called a triad. Okay, so it's made of three things, two terminal cisternae and a T-tubule. That's called a triad. 1, 2, 3, triad. So each myofibril is made of hundreds to thousands of myofilaments, which consist of one or more of the following types of proteins, which it's more. So you have contractile proteins that are going to be generating tension, eliciting a contraction. This makes sense. Regulatory proteins that dictate when a fiber may contract. They're going to regulate it, literally, regulatory proteins. And structural proteins that maintain proper myofilament alignment and stability to support the structure. Makes sense. All right, so three types of myofilaments. Again, just talking about essentially proteins. Okay, so thick filaments are composed of bundles of the contractile protein myosin. Thin filaments are composed of proteins actin, tropomyosin, and troponin. If you work in the medical field, you will know that this troponin sounds very familiar. Okay, so typically associated with the medical field, troponin, that's your cardiac version of troponin. We're specifically talking about skeletal muscle troponin, but it's roughly the same thing. You also have elastic filaments, which are composed of a single massive spring-like structural protein, which is one of my favorite ones. It's called titin. That's going to stabilize the myofibril structure and resist excessive stretching forces. Again, maintaining that structure. Okay, so while we're on these proteins and everything, let's talk about Duchenne muscular dystrophy, DMD, which is honestly how it's usually referred to. So DMD is a degenerative muscular disease occurring almost exclusively in boys. So it's caused by a defective gene for the protein dystrophin, which is coded for on the X chromosome. So we said this was almost exclusively in boys. Wait a minute, boys are XY versus females are XX. Why does that make sense? Well, this is coded for on the X chromosome. So essentially, boys only have one X and again one Y. So if you've got a dysfunctional X, that's the only one you're going to get. Versus females, if you have one, so to speak, bad X and one, so to speak, good X, that good X that's making that protein, a functional protein, you can use that and not have this. Versus in boys, again, you only have one X, the other one's a Y, that's what makes you a boy. You know, chromosome speaking. If you have one, so to speak, bad copy of that X, you're going to have this. Okay, so that's why it makes sense that this is more common, almost exclusive in boys. So dystrophin, again, is a protein, and that's found in striated muscle fibers, and it's going to anchor the sarcolemma to the surrounding connective tissue and to the myofibrils. Okay, so essentially in DMD you have fat being replaced, muscle being replaced by fat. So this is what your normal muscle fiber would look like. Okay, normal. Versus in DMD, all that is replaced by, again, fibrous tissue or fat. Okay, so again, once you replace something with either fibrous tissue, fat, whatever it may be, that tissue is not going to be as adequately functional anymore. So this is extremely debilitating. So in the absence of normal dystrophin, the sarcolemma breaks down and the muscle fiber is destroyed and replaced with fatty and fibrous connective tissue. So symptoms typically arise between the age of 2 and 12 years old, typically younger, and they include weakness to the proximal limb muscles and typically a waddling gait if they're able to walk at all. And generally, they're all wheelchair bound by the age of 12 and dead from respiratory or cardiac failure between the age of around 20 to 30, it depends. So again, why does this make sense? Your diaphragm is a skeletal muscle. It is striated, so respiratory failure, that makes sense. Cardiac failure, your cardiac muscle is striated as well, so it's going to affect that muscle as well. It's going to replace some of that functional muscle with fat, and over time, it's going to add up again. Typically, it's fatal between the ages of 20 and 30. All right, so let's talk for just a second about the myofibril. In these next few slides, we're going to talk about specifics on thick and thin filaments, and generally the myofibrils as well, sarcomeres and stuff like that. All right, so thick filaments are composed of the contractile protein myosin. So remember the name because we're going to come back to it. It's going to be very important when we talk about muscle contractions because it's a literal contractile protein. So each myosin has globular heads at each end linked by intertwining tails. So the myosin heads are connected to the tails by a hinge-like neck. Okay, so each myosin head has an active site that will bind with actin. So the love story between myosin and actin is one of the best ones ever. I love talking about muscle contractions. We're going to talk more about these in just a second. So, speaking of actin, thin filaments are composed of actin, tropomyosin, and troponin. So the actin subunits, seen here in purple, are together going to form the two intertwining strands in the functional thin filament. So one string of pearls here, and another string of pearls here, or purple beads, whichever one you think about. All right, so each actin has this kind of like indentation here. It's called an active site, and that's actually where this myosin head is going to bind to this active site. Okay, so again, you also do have, let's see, yeah, it goes on to it in the next slide. You also do have tropomyosin, which is this kind of blue portion here, these blue strands. These are rope-like regulatory proteins that are going to twist around actin, and if you'll notice here, they're all covering those little indentations. It's actually covering this active site. So when this tropomyosin is here, that myosin cannot bind. So you also have another regulatory protein called troponin, this yellow portion here. It's a small glandular regulatory protein that holds tropomyosin in place, and when calcium is bound to troponin, it will assist with turning contractions on and off. So if troponin and calcium bind, this will actually have a conformational change, and this tropomyosin actually shifts, and these active sites are open. Therefore, it would be able to bind with myosin and elicit those contractions. All right, so this next portion is talking about connective tissue, which we talked about in, I think it was chapter 9.1. I'm just going to run through this real quick because we've, again, already gone through this, but I do need to go through it. All right, so superficial to the sarcolemma that surrounds each myofibril is the endomysium. It's this extracellular matrix that bundles several myofibrils together. So the endomysium plus the myofibrils is essentially a muscle fiber. So several muscle fibers are bound together by the perimysium. Perimysium plus muscle fibers are what you call a fascicle. And again, this is nothing new. We already learned this. We learned this in the lab. We learned this in 9.1, and there's a really good picture. I think it's coming up here. Yeah, really good picture showing you this. And bundles of fascicles that make up a skeletal muscle, which is surrounded by the epimysium, making up the whole of the skeletal muscle. Okay, and that's just telling you from smaller to larger. And that's, again, just putting it in relation of those connective tissue wrappings. And again, that muscle will taper down at the end to form this this tendon. This connective tissue will essentially attach the muscle to the bone. Again, that's what tendons do. They attach muscle to bone. All right, so the perimysium and the epimysium come together at the end of the muscle to form a tendon that binds the muscle to its attaching structure, which is a bone. Skeletal muscles are enclosed by a layer of thick connective tissue called fascia, which anchors them to the surrounding tissues and holds groups of muscles together. Just telling you the same thing. All right, so striations. Well, let's talk about striations for just a second. So striations appear microscopically as alternating light bands and dark bands. So that's actually what you saw when you looked underneath the microscope. You can see these little like lines. They almost look like little lines on like a piece of salmon or something. Makes sense. Okay. So they're little alternating lines between light and dark bands. Okay. So light bands are where only thin filaments are found, and dark bands are where both thin and thick filaments are found. Okay. So light areas of striations include the following specific regions. The eye band is only composed of thin filaments. So light bands are eye bands, so to speak. The Z disc, which is in the middle of the eye band, is composed of structural proteins that will anchor the thin filaments in place to one another and serve as attachment points for elastic filaments. They also attach myofibrils to one another across the entire diameter of the muscle fiber. I know it's pretty wordy here, but I'm going to show you a video at the very end, and it's going to hopefully make a lot more sense once you can see all of this moving and how everything works. Hopefully that'll make a lot more sense to you. This is, I know, kind of wordy, so bear with me. All right, so dark bands are called A bands. They contain the zone of overlap, the H zone, and the M line. So the zone of overlap is the region where we find both thick and thin filaments, and this is where tension is generated during contraction. Okay, that's pretty important. The zone of overlap is the region where you have thick and thin filaments, and that's the area where tension is generated during contraction. Okay, so that would be right here. And that's really what we're going to focus on in a little bit. Okay, so the H zone is where only thick filaments exist, and the M line is structural proteins that hold the thick filaments in place and serve as an anchoring point for elastic filaments. All right, so let's talk about the sliding filament mechanism of contraction. So the sarcomere extends from one Z disc to the next. It is the actual functional unit where this contraction occurs. So the sliding filament mechanism explains how tension is generated during muscle contraction. So during a contraction, both the I band and the H zone will narrow, while the A band remains unchanged. Myosin heads attach to actin and pull the thin filaments towards the M line, which brings the Z disc closer together, shortening the sarcomere. So sarcomeres are arranged end to end within each myofibril, and when simultaneously contracted, they shorten the whole muscle fiber. So one thing you need to remember, okay, even though they're going to shorten, this does not mean the filaments themselves actually shorten. So what really happens is the thin filaments just simply move towards the M line. Okay, so this is essentially what happens in a relaxed sarcomere versus a contracted sarcomere. Okay, so relaxed versus contracted. Okay, so if that didn't make sense to you, if that's a lot of really wordy stuff, I have 2 different videos that would help you. I have a simplified version and I have a little bit more detailed version. So all you have to do is click on these. Again, we're going to talk more about it, but if that didn't really make much sense, I do have help for you.